Time to start the afternoon session. The first pair of talks are on therapeutic neuro ultrasound. And they're a little bit inhibited, but the first speaker is a good friend of mine, Jay Sanguinetti. He is from University of Arizona and also San Mai, his company, and he's going to talk about exactly about ultrasound. But what's your title? Really? Untangling neural correlates of consciousness with ultrasound. Oh, there it is. Jay Sanguinetti.
All right, thank you, Stu. As he said, I'm going to talk about untangling the neural correlates of consciousness with brain stimulation. Most of this work was done at the Center for Consciousness Studies in collaboration with Stuart Hameroff and others, as well as the Department of Psychology with John Allen, who's in the department. And as Stu said, I do have a company as well called San Mai Technologies Public Benefit Corporation, which is working on similar technology that I'll talk about today. Starting with the grandfather of psychology, William James, who said, "Our normal waking consciousness is but one special type of consciousness. Whilst all about it, parted from it by the flimsiest of screens, there lie potential forms of consciousness entirely different." And so, of course, William James was writing about consciousness and the science of consciousness over 100 years ago. What's that, 103 years ago? And we can ask the question, how much progress have we made with the development of neuroscience and all the tools that have been developed with neuroscience, as well as modern medicine and modern psychology? What progress have we made in the last 100 years? Obviously, this conference was developed because you know the answer, and we're calling for a revolution in that. In science, we look for something called consilience. So I'm not going to make you do this, but if I had you stand up and you all jumped together, that would be consilience. In the science of consciousness, we don't have a lot of that. So if I asked you to pick your favorite neural correlate or your favorite piece of the brain or your favorite oscillation, you'd all be jumping at different times. And that's a problem if we're really looking for progress in science. Our evidence and our ability to make inductions and conclusions should be converging towards a central theory of consciousness. And that leads to questions. For example, questions that Stuart asks about what's going on. Are we missing something fundamental about the way that we're doing our science, about where we're looking in the brain or the body, or perhaps how we're looking at the physics in general? Now in neuroscience, we have decided mostly to look for the neural correlates of consciousness. So of course we have to use the tools at our disposal and the methods that we have in science at our disposal. And so as most of you know, the neural correlates are one way to enter the avenue of consciousness studies for neuroscience. We're basically looking for the minimum set of neural mechanisms that are sufficient to produce a conscious percept. Understanding that at this point in time, we're not going to be able to solve what's called the hard problem of how these neural mechanisms are actually causing these things to happen, but more looking at the correlational nature. And if you look through the papers, as I've been doing over the past 15 years, you'll find all kinds of neural correlates. So this is where if I asked you to jump, you'd jump for different things, from brain regions to brain rhythms, like gamma binding, for example, all the way through to connectivity, looking for feed-forward and feedback connectivity, and now there's the predictive coding model. All of this has led to many different theories about consciousness, so much so that the Templeton Foundation funded something called an adversarial science project that Dr. Hameroff was a part of to try to get these theories to compete, to try to falsify each other. And what we have so far is not a lot of consilience going on in the science. There's lots of healthy debate, and that's obviously fine. That's what science does as well. But the problem is, over the last 100 years, you would hope some convergence is happening. And actually, we're in a situation where other really interesting theories, like OR, for example, we don't actually have the current tools to distinguish between these and try to start falsifying some of these different theories to let us understand if some of them are sort of better than others. And so why is there so little consilience? Well, as Stuart would say, maybe our fundamental assumptions are wrong. Maybe we're looking in the wrong place, for example, or we're using the wrong tools. are perhaps the tools that we're using are not actually allowing us to distinguish appropriately between the pieces of evidence that are coming in. So for example, with functional magnetic resonance imaging, or fMRI, we're essentially looking at correlations between brain activation, so where the brain is lighting up, and conscious states or contents. Now this has been the basis of most of the neural correlates of consciousness, using either fMRI or EEG, as Anirban was talking about. The problem with this is that with these methods, we actually can't distinguish between these three things on the screen. So are we looking at neural prerequisites for consciousness, meaning things that are necessary but not sufficient to cause it? Are we actually looking at the brain lighting up for neural substrates, so the things that are necessary and sufficient? That's what we're looking for. Or neural consequences, so you have brain activation that's related to a conscious percept, for example, and then the brain activity you're seeing is the consequence of that action, not actually the cause. So with the current correlational methods, we don't really have a way to disentangle these three issues. And if you look in a lot of the papers, the word that most people use is neural substrate. So most people are claiming the brain activation that they're showing, for example, in this study, is a substrate when in actuality it could be a prerequisite or a consequence. This is another way to say what I just said. So with functional imaging methods like neuroimaging, it's hard to tell if the region that we're looking at is actually functionally relevant for causing consciousness or the conscious states. And so we need other methods. For example, methods that could causally disrupt the supposed substrates. Then you could look at or measure the effect on consciousness or its contents. Then you would have a better way of knowing whether what you're looking at is actually causally related. So for example, Wilder Penfield in the 1940s did some classic and famous studies on patients undergoing epilepsy. So he was mapping brain regions to make sure he wasn't removing functionally relevant brain regions when they were doing surgery. And they found that they could reliably produce certain types of conscious percepts. For example, one patient would smell their grandmother's pie. And every time they touched, let's say, number 11 there, they would get that same type of experience from that patient. Now, obviously, these are pretty crude methods, but it shows the power of being able to causally manipulate or disrupt brain activity. So we want to go from those types of tools to more precision. So if microtubules actually have something to do with conscious states, we would want to have tools that could causally manipulate those microtubules, and not damage them, of course, but manipulate them, disrupt their function, and then look and see if we have effects on consciousness. Stuart's talked about anesthetics as one way to do that. Potentially brain stimulation is another way. And so back in 2015, Stuart and I were talking about these results that Anirban was showing, where he found that microtubules have resonant frequencies between kilohertz and megahertz, so somewhere between 12 kilohertz or 12,000 oscillations a second up to 30 million oscillations a second. Then Anirban published some really compelling studies showing that microtubules in a dish, so these aren't in the brain, but he had created microtubules in a dish and showed that, I think I have the thing here. So you see this microtubule in green. He's got essentially like a patch clamp. It's a pretty interesting method that he developed. And what you're seeing in the graph there is actually at different frequencies, he could disrupt the resistance to the microtubule. So it's disrupting its electrical properties, showing the resonant vibrations there at 10 and 20 megahertz. So this suggested that microtubules could be disrupted or influenced by megahertz frequencies, which is what ultrasound is, and led us to start thinking, or actually led Stuart to start wondering, can you put an ultrasound on someone's head and modulate their conscious states if microtubules have something to do with consciousness? Turns out that there's a whole field that was developing transcranial ultrasound as a way to stimulate the brain that actually dated back to the 1920s. And the basic idea is that you could focus a beam of acoustic energy, usually at about 500 kilohertz, into the brain. And you can essentially focus that anywhere into the brain and use mechanical vibrations to disrupt the electrical or other properties of the cell, causing brain stimulation or brain inhibition in different methods. Now there is a whole lot of studies coming out about this now, but when Stuart, I think, was maybe one of the first humans to put this up on his head in maybe 2012, 2013?
2012, I think, yeah.
Yep.
My colleagues called my bluff. You want to try it on patients? You go first.
Yeah, so Stuart was in the hospital, as you can see in this picture, and he was thinking about this hypothesis, thinking about ways to try to falsify his own theory, and wondering if microtubules could be resonated with ultrasound to change consciousness. And I think the story goes, he tried it in the hospital. You felt like maybe it was like sort of drinking a martini or something like that.
Nothing happened for about a minute, then I got buzzed for about an hour and a half.
And then you didn't go back to work, right?
It was the end of the day.
Okay. So then Stewart followed this up on chronic pain patients to find out if he could modulate their pain perception and their mood. So he put it up on the right prefrontal cortex and actually showed that although chronic pain wasn't significantly reduced, their mood perception was, which is actually quite hard to do in pain patients if you know them. So Stewart approached our group. I was working on deep brain stimulation at the time and asked if we could replicate this outside of his lab. So when I was a graduate student, we started replicating these effects, targeting parts of the brain that deal with mood and emotional control, called the right prefrontal cortex. And basically over about 300 subjects in a couple different studies, we were able to replicate these effects, essentially showing that ultrasound to a mood circuit in the brain can modulate mood. Right, so that was like one of the first two reports where human studies are showing that ultrasound is affecting mental states in this case. And here we also looked at fMRI, so we looked at brain activation as well, and showed that the brain changes were correlating with the changes in mood. So since Stewart bravely put the ultrasound up to his head in 2012, lots of human studies have now come out showing that this is a quite powerful method to modulate brain activity. So Sunshik Yu's group at Harvard tried to use ultrasound to modulate the visual cortex. So obviously we know that the visual cortex is related to visual perception or visual consciousness. And what they did is they put a little beam of ultrasound right into V1 or V2 of the visual cortex, and they were able to stimulate visual phosphemes in the majority of their participants. So basically they've got an ultrasound in the back of their head and they're looking into a dark room, and every time the ultrasound turned on, this is the visual experience that a person would have. Each person's experience is a little different. Some people get geometric shapes, some people get light flashes, but everyone was reporting these phosphemes. And importantly, they also showed that you got visual evoked potential, so it was very reliable evoking of the visual response. And finally, we started wondering, can we use this method to modulate deeper structures in the brain that are involved with self-perception, self-consciousness, and really sort of more in line with research in the meditation and psychedelic space, which is what our lab is really interested in. So we started looking at something called the default mode network. You're probably familiar with this. It's a network that comes online when you're mind wandering. So right now, about half of you are in your default mode network and not listening to me. Now maybe 80% are listening for a minute. So the default mode network is a crucial network for self-processing. So you think about the past, you think about the future, you're planning, you're going over smart things you said, dumb things you said during the retreat or during the conference. It's very important for self-processing, and it's modulated both by psychological disorders like PTSD or depression, as well as when you do things like meditate for a long time or take psychedelics. And that's because this network that I'm showing on the screen seems to be coordinating all the different networks in the brain that are relevant for self-processing. And this is a really great paper by Andrews Hanna, who's actually at our university as well. So for example, if I took you in the lab and I gave you psilocybin, magic mushrooms, and I gave you a really big dose, this is the type of brain activation change you would see. And particularly right in the back here, I don't know if I have a laser. Yeah. So this back part here is the major hub of the default mode network. It's called the posterior cingulate. And the more psilocybin you take, the more that region is going to deactivate. So deactivation then relates to extremely intense effects, leading to mystical effects in these patients. These were people undergoing psychotherapy as well. And if you get to a threshold, you get something called drug-induced ego dissolution. They call it DIDE in the literature. This is the ego death. That is basically the subject is reporting a very significant mystical experience that then predicts actually health outcomes later in time. And the important point is that it's really related to disruption of the DMN. However, as I was pointing out earlier, with these methods, it's hard to tell if that's causative, right? I can't say with this correlational method that that's causing that mystical experience. It could be a byproduct of it, for example. And so my colleague at Brown, Judd Brewer, and his group actually tried to tie directly the posterior cingulate activation to phenomenology. So they said, if this is really related to the moment-by-moment change in perception of self, we should see these changes in time. And so they created an fMRI neurofeedback paradigm. So these participants are seeing this line in real time in the scanner, and they're trying to meditate. And basically, they're tapping in to see what their phenomenology is like throughout this 30 minutes. And what they found is that long-term meditators who are meditating could significantly reduce their default mode network activation relative to baseline as they're meditating. And you can actually see they're sort of sinking into the meditation throughout this time, whereas controls had the opposite effect. If you've ever tried to meditate, the first couple times it's pretty hard, right? So this, again, is related to the activation of this area. And these meditation folks were actually reporting pretty deep states in the scanner. Meditators like to be in the MRI for some reason. So they're having somewhat like mystical states. And this actually developed a hypothesis in our lab that, all right, if this is really the case, that this posterior cingulate hub of the default mode network is related to deep levels of self-perception and really organizing self, then we should be able to temporarily disrupt it with focused ultrasound in the scanner and produce these same type of effects that we're seeing in both the psychedelics and the meditation literature. So back during the pandemic, we were the only lab at the university allowed to run human subjects. We brought them in and we put them in the scanner. And we targeted the default mode network, the posterior cingulate, in each of those participants. And we asked a basic question: can we change the network connectivity? And does that change predict phenomenological changes like we see in the other studies? Oh, I just pushed the wrong button. There we go. So we found overall that we could change default mode network connectivity relative to placebo. So basically everything you're seeing in blue is a change in connectivity, and the DMN, or the default mode network, is that sort of yellow line there. Importantly, when we tap in with participants, we ask them, what's this like for you, right after this. Basically what we find is that relative to placebo, we got a significant change in questions that deal with like inner peace, sense of time, sense of ego loss, which we weren't predicting too much of that, merging with the surroundings. And importantly, we weren't expecting them to get more paranoid, more afraid, fear of losing their mind and things like that, because that would be a different part that I could stimulate to do something like that. And basically the way to read this, sorry I didn't explain it, this is 0 in the middle, and this is more intense as you're moving out. So what you're looking at is sort of the phenomenology map of these participants, and clearly you can see in the blue line that that was changed relative to placebo. So overall, we replicated what is coming out of both the psychedelic and the meditation literature in the sense that the posterior cingulate hub really looks like it's coordinating activity related to sense of self that then relates to things like inner peace that you would expect if you're disrupting or loosening up that system. In our lab, we're really interested in a concept called equanimity, so the ability to be open to yourself and your states, pain and pleasure. And I'm not showing you this data, but the equanimity effects are actually predicting some of these effects as well. So we think we're loosening up the self-structure in an appropriate way to allow people to have these changes. So far, we're seeing that ultrasound modulates mental states in both Dr. Hammeroff's studies and ours, as well as perception and self-awareness. This is leading to causal evidence for neural substrates. However, these are brain regions. And in my view, pointing out different brain regions or mapping them still isn't going to get us too far to places like where Stuart wants to go, which is what's the functional nature of the processing units, right? So we're just kind of mapping the brain regions. It's still pretty crude. And then we could ask questions, you know, what about the microtubules? So am I showing evidence that microtubules are really involved in consciousness? Well, sorry, Stu, not quite so far. The reason for that is when you look at what is the ultrasound actually doing to the brain, what are the mechanisms, how is it influencing the activity or the processing units, both in the neuron or inside the neuron. Most of the field so far is going to recognize these next two. So ultrasounds affecting ion channels. We know they have a stretch sensitivity. There's actually a piezo 1, piezo 2 ion channel that's been discovered recently. So the mechanical vibrations could be affecting the channels. It could also be affecting the electrical properties of, for example, the lipid bilayer by actually moving, right? We're talking about 500,000 oscillations a second going across the brain. You're actually moving the membrane and oscillating it in ways that changes its electrical properties. And, of course, the resonant hypothesis for microtubules could be happening as well. And so I think the job for us now is to try to separate out these three mechanisms and try to figure out, can we hit the resonant vibrations of the microtubules in such a way that we get changes in microtubule resonance without changing ions or these sort of vibrations across the lipid bilayer. It's quite possible to do that if we do really short bursts of ultrasound, for example, or maybe use an intensity level that's too low to affect these mechanisms but still hit the resonant frequency. Like if you pluck a guitar string, you can pluck it, if you hit the resonant vibration, you can pluck it with a very low amount of energy and get the resonance. But if anyone else has ideas about how to separate out these mechanisms, we would love to hear them. So what can we conclude so far? Well, the neural correlates of consciousness have obviously been important in neuroscience in general, but we're not making as much progress as we want. And that's likely due to the limitations of our methods. To advance the science, we need to move to the neural causes of consciousness, so I'm going to give that a little c for cause. And that hopefully will allow us to look for more causal evidence about at least the brain regions, maybe ultimately getting to the brain processes underlying consciousness. And transcranial focused ultrasound is an ideal candidate for studying these neural causes of consciousness because of its ability to focus into the brain, into specific brain regions. And then we can measure those causal effects that we're looking for. However, I will give the caveat, there's still a lot of issues with this, particularly getting the ultrasound across the skull. And so there's many groups trying to work on the specificity of ultrasound, getting it into the brain, and really trying to understand the mechanism, as I said. So really, if we want to get to trying to falsify the microtubule hypothesis, which I think Stuart is into, then we need to try to separate out those mechanisms so that we can hit the resonant vibrations of the microtubules without affecting any other parts of the cell. So if you have ideas to do that, please let us know. Maybe Sterling will tell us next how to do that. Okay, and these are all the folks that have been working on this research, including Dr. Hammeroff, Dr. Allen, and Shenzhen Young at the Similab, and Vince Clark. All right, thank you.
Thank you, Jay. We have time for a couple of questions. Daniel.
Yeah, just a quick question. Are the resonant frequencies of the membranes and the microtubules, are they the same? I thought microtubules are stiff, kind of rigid things. Would they have higher resonant frequencies well beyond the 500 kilohertz range?
You want to answer that?
No.
I don't know the answer.
Could that be a way to separate them? I mean,
I know Anirban found different resonant frequencies depending on...
That's right. So if you can separate them out by the resonant frequencies, that'd be the simplest thing to try.
Yeah, and then hit that resonant,
Yeah.
Yep.
Let me just say that if it's the membrane ion channel, a lot of those are metabotropic, so they're connected to the cytoskeleton. Do you know if those particular ones are connected, like through G proteins? Because if you vibrate them, they're going to transmit the vibration into the microtubules. Although I would think the ultrasound will go right through the membrane anyway.
Yeah, it'll go through.
And one other point, Anirban did a study on just pure microtubules, no cells, no membranes, and looked for the resonant frequency that caused the most polymerization with electromagnetic vibrations, not ultrasound. And I think 10 megahertz was the optimal. Is he here? I think he's probably...
He's doing the experiment.
But 10 megahertz electromagnetic made optimal polymerization states. But one way to approach what Jay's talking about is to do studies at the cellular and microtubule level.
This way? Very nice. One thing, with the talk about now that we're doing fMRI and a lot of non-invasive techniques, one thing that's being forgotten in terms of looking at correlates of consciousness is lesion studies. Mm-hmm. And we know, and it tends not to get mentioned, we know that patients with lesions have alterations in consciousness, they have reduced self-awareness, they can have reduced awareness of illness, reduced awareness of who they are. So I think that is, it's not going to get at the microtubules, but it may get at the global, more not macroscopic, but more global causes of consciousness in terms of brain anatomy.
Yep, totally agree. I took that slide out because we only had 20 minutes, but there's a researcher at Harvard, look up Neuro Michael at Harvard. He's doing some really cool work with network connectivity analysis, so that's this global network analysis for lesion databases. So they have these giant databases now, and they can look at how different changes in networks change conscious states. And he's actually found a spot of the brain that's really deep in the brainstem called the periaqueductal gray area that relates to mystical experience. And it's like one of the oldest parts in our brain. It's totally unexpected.
And you see, the microtubules have a role. If the microtubules have a role, and they may well have a role, I'm not saying they don't, that role will interact with where the microtubules are in the brain. So the microtubules in the frontal lobe may have a different effect than affecting the microtubules in the parietal lobe. So I think you do have to take into consideration the anatomy.
Mm-hmm.
I wouldn't disagree with that, but they can also be entangled, and it seems like it spreads too. Do you think that with focus that it spreads, or it just stays in one place?
The beam?
Yeah.
Definitely spreads, yeah. I mean, there's the focus that you saw. There's the focus that you were seeing, but the beam keeps moving, and it hits off the back wall of the skull and moves back. So, yeah, it's definitely moving around.
I prefer unfocused because you can't do the kind of studies he's doing, but I think for therapy it might be better and simpler.
Right, but even though the ultrasound is unfocused, what's happening in the different parts of the brain in relation to the microtubule?
Yeah, but if it bounces around, it's going to hit all those places anyway.
That's right. But how the microtubules are affecting consciousness, they may be doing something different in different parts of the brain. Sure, you'll be able to hit them all, but if you really want to know what's going on, you got to know what the microtubules are doing in the different parts, even if they are entangled. Mm-hmm. It may be different. There's an interaction, is the way it seems to me.
Well, if you ask me where, I'd say, thank you. If you ask me where, I'd say cortical pyramidal cell dendrites and cell bodies, because they have the largest arrays of mixed polarity microtubules. Okay. That's where I'd bet consciousness.
But that's testable. That's testable.
Yes.
I think we got to move on time-wise, so you want to?
A very quick question. You showed the phenomenological plot with the placebo in the ultrasound. How close did psilocybin correlate with what you plotted for the ultrasound?
That's a good question. So we're more on the order of microdoses. I usually don't show that because I don't want to make it look like we're trying to recreate psychedelics with this. But yeah, it's like a microdose type effect. We want to be careful because these are, consenting undergraduates in our study, so we don't want to like make it look like, big dose of LSD or something.
All right, let's thank Jay for a great talk.
Thank you.
Well,